Okay, hi, hello everybody. I'm very delighted to have our guest speaker today, Professor Serge Picaud. He's currently the director of the Vision Institute in Paris and is one of the founders of this institute along with Professor Sahel. He's heading also the team of the retinal information processing at the Vision Institute in Paris. Professor Sahel did his PhD in University of Marseille and then a postdoc under the supervision of Professor Werblin in Berkeley. He was a research associate in Max Planck Institute of Brain Research, working along with Professor Wassel. Professor Sahel work is trying to understand the normal.
I'm Serge Picaud. Picaud.
Huh?
Professor Picaud.
Sorry. Professor Picaud is currently, the team is working on the physiology of the photoreceptor and the synaptic output, the glutamate transporter, and working on the input signal, the GABA and the glycine receptors. He also characterized some of the drugs that are associated with GABA inhibitors and discovered the importance of taurine for retinal ganglion cell, and this has some clinical relevance. Recently, he's working more on the field of vision restoration, both in the field of high-resolution retinal implants, 3D retinal implant, as well as working on advanced restoration by optogenetics, working along with gene site biologists. And Professor Picaud is well known for more than 150 peer-review publication, numerous patents, and we are very welcome to have you here. And thank you for agreeing to talk. Thank you.
So thank you a lot, Yossi. I mean, it's a great pleasure for me to present this work, and I hope we will have a very fruitful exchange. So the work is on vision restoration because, in fact, not much thing has changed since Peter Bruegel, and so in the 16th century. And so we want to try to improve the autonomy of patients. And so as we can see here, vision loss is among the most feared health issues. And not only it has the greatest impact on daily life, but also people are ranking it at the worst condition. So in fact, many diseases can induce blindness. We can see on the right that in fact, the number of blind people will double until 2050. And blindness, I mean, is due mainly to disease. Of course, there is cataract in low-income countries, but we have also age-related macular degeneration, AMD, and glaucoma, which are leading to many blind in our countries. So the idea is to see if we could restore vision. So what we have been doing mostly was to work on patients losing photoreceptors. And as you can see, in fact, when patients are losing photoreceptors, we still keep in the eye, in the retina, 2 layers of neurons. So the idea was to see whether we could reintroduce some vision into this retinal circuit. And the first ideas was to put some electrode arrays either in the subretinal space or epiretinal in the vitreous. And so the activation of electrodes would induce or generate some activity in these remaining neurons so that some visual information would leave to the brain. And so the patient with photoreceptor degeneration are mostly age-related macular degeneration And you see these patients, they're losing central vision, but they keep this peripheral vision with a quite low visual acuity. So it's why these patients were not included at first in the clinical trials, but instead patients were included patients were having retinitis pigmentosa, where they lose peripheral vision, and then they lose also this pericentral tunnel vision. So the first retinal prosthesis was generated by the company Second Sight, and it was named Ergus 2. And so as you can see, the patient were wearing goggles like these, taking images so that these images would go to the microprocessor, and then the images will be converted in codes for electrical current. They would be sent from the emitter to the receiver, and then this box is containing a chip that will generate the current diffusing from the chip to the electrode array that is nailed onto the retina. And so it requires that there's an opening in the eye for this wire to get to the electrode array. So we see that this electrode array contains 60 electrodes. And of course, the patient with this electrode, they can point to a square on the screen when the system is on. When the system is off, of course, they cannot. And then this is very stable. As you can see, up to 4 years, there was no loss of this restored vision. So, but with this implant, in 60 electrodes, this is at best what you can get. I say at best because in this case, all the electrodes are working, and also they are providing gray levels, which might not be the case in this electrode. And especially, I mean, most of the patients did not get 60 electrodes working at the same time. So you can see with these images that it's quite difficult to recognize the face with 60 electrodes, but already with 600 electrodes, it's possible to recognize the face. And this was shown that, in fact, with an electrode array with 600 electrodes, you may have independent locomotion. You can start to read text and even complex text. So it would be possible to have some kind of restored autonomy. So this is more or less the aim that all the team have looked for. And so this is also what was done by Retina Implant AG, a German company. And this company has generated a chip where you see, in fact, there's 1,500 electrodes. So this chip is here under the retina. So it's a subretinal implant. In this case, you see that the chip is here connected to a wire. And this wire, in fact, is getting back at the back of the ear to be connected to a battery. Because in this chip, there's a circuit that needs current to amplify, in fact, the current obtained from the photodiodes. So the surgery for these implants was very long. And in fact, also the wire is establishing some kind of stress on the electrode array, so that sometimes it was also moving. And in fact, this is with this electrode array with 1,500 electrodes, the patient are doing, can recognize, I mean, some patient can read letters, but it was the same with the Second Sight array. And in fact, the performance of the patients were not much better than with Second Sight. So we may wonder why. And in fact, Jutla and Iber has proposed an explanation. So what they showed was that in this electrode array, you have electrodes. And in fact, the current has to return to a very distant ground. And so if you model the current level in the tissue, you see that at the stimulation point, the current level is not highly different from that in the neighboring tissue. And if you want to increase this difference, you can use different types of configuration from concentric bipolar to ground surface. The idea is that if you, in fact, include the stimulating electrode in a ground grid, it can really induce a big difference between the stimulated area and the neighboring area. And this is especially true if the ground grid is highly conductive. So this led us to look for highly conductive materials, and it's why we have worked on graphene, especially graphene electrodes. And we could show that with graphene, we can use this material for recording, and you see here spikes of ganglion cell. We can even record light response of the ganglion cell. You see here when we stimulate, we have an increase of spiking frequency at the on and off set of the light. But also what we showed was that when we use some electrical current, when we stimulate with these graphene electrodes, and you see here the artifact of the stimulation, we can generate spikes in response to this stimulation. So graphene is quite nice, not only to record, but also to stimulate electrodes. And we could show also by having an implant here with covered with graphene that there is no immune reaction to the graphene implant, I mean, compared to polyimide controls. So this suggests that graphene is a highly conductive and also highly biocompatible material. So we are moving on this, but what I would like to show is some result that we gained with retina implant that was generated by Daniel Palanca. And the novelty of this retina implant was that they contain a central electrode, but also a ground grid here that is surrounding, in fact, the stimulating electrode. So all this honeycomb pattern is, in fact, a ground grid. And what you have here in red are photodiodes, infrared photodiode, so that, in fact, this implant is completely wireless. So you introduce this implant below the retina, and the stimulation of the photodiode will provide activation of this electrode here, and also will provide the power to activate the electrode. So it's quite nice. So this implant was generated by the team of Daniel Palanca, and Yossi worked a lot on this implant. So what they showed was that if you introduce such an implant into such a recording electrode array here on a blind rat retina, you can in fact stimulate the implant with infrared such that it will generate spike in the retinal ganglion cells. And then what they did was to move to in vivo rats, where they were measuring visual evoked potential and stimulating the implant with stripes so that they could measure, in fact, the visual acuity using this electrode array. And they could show that, in fact, the visual acuity with this type of implant is not too far from the normal visual acuity of normal rats. So these implants were very interesting, and we decided with Pixel Vision, our startup company, to, and of course with Daniel Palanca, to produce this implant in France. So the question was, can we in fact can we produce this implant at a clinical grade and demonstrate that they can activate the primate tissue? And so this was our aim here in Paris. And so we obtained some primate retina, but of course there's no blind primate. So we generated a model of blind primate by simply shaving off the photoreceptors with a vibratome. So we are generating a section of the retina in its thickness so that we could get only the inner retina. And then we were recording, as Daniel did, in a multi-electrode array, the ganglion cell here, while having the implant above the retina, stimulating with infrared the implant so that it would release some current, and so define whether we were able to activate the retinal ganglion cell from a primate retina. The idea is that primate cells are much bigger than those from rats, so it was not evident that we would be successful. When we did this, we could show that when we stimulate, we see the artifact, and we do see spikes in retinal ganglion cells. And what we were very pleased was that when we were stimulating other units than this pixel 60, we had no spikes from these retinal ganglion cells. And you see here, we had stimulated this unit, but we also stimulated all the neighboring ones. And you see that only this single unit, pixel 60, was able to activate that retinal ganglion cell. So we could show that, in fact, this implant can have a very high resolution. And so then we moved to in vivo. So each unit here was 100 micron in diameter. So when we move to in vivo, you see here, we can introduce the implant below the retina of living primates. I think it's not visible enough on this OCT, but in fact, what we could see is that the photoreceptor layer is disappearing, simply because this implant is in fact separating the retina from the choroid. And you see here on a histological preparation that in fact here in green, you have the cone photoreceptors, and you see here at the position of the implant that the cone photoreceptors are degenerated, whereas they are present just neighboring. So in fact, we are creating a blind spot here. So what we did was to test with a behavioral test whether we could elicit some activity in this implant. So what we did was a test where we asked the primate to generate a saccade. So we first asked him to concentrate on a central point, and then we would present a peripheral spot and asked him to generate a visual saccade so that he would tell us that he had seen this peripheral spot. So of course he was generating saccade in all directions, except here at the position of the implant. But if we would stimulate with infrared, we would see that he would generate a visual saccade node due to the stimulation of the implant. And what was nice was that he was not generating visual saccade in infrared to any other direction. So this was clear that it was the infrared activation of the implant that was allowing the visual saccade. And what was even better was that stimulating only one single unit was sufficient to generate a visual saccade. So then we moved to a patient, and so this was generated by the company Pixel Vision. And you see here the patient. The patient was able to read the letters, and now they can read words. And recently, so the visual acuity measured for this patient was between 20/460 to 20/565. This is a paper by Palanca and Sahel. And also, more recently, what was generated was some goggles like these, where the patient can still see with natural vision in the periphery and merge the performance with the artificial vision in the center. So you see here, in fact, the implant, and here the projection of the image on infrared by infrared on the retina. This is very nice, but you see that the visual acuity is reaching now the level of legal blindness. But we wondered whether we could further improve this and try to reach cellular resolution with an alternative technique. And this alternative technique is optogenetic therapy. So I guess that you may all know it. The idea is that in algae, there are some channels that are light sensitive, like channel rhodopsin. And so the idea is just to take the genetic code of this photosensitive ionic channel, to put it in an AV, inject the AV in the eye so that retinal neurons will express this photosensitive channel and will become photosensitive. So we kind of reintroduce some pseudo photoreceptors in the eye. So this was done at first by Zhao Pan in Detroit, where he introduced the channel rhodopsin in ganglion cell, and he could show that he was able to induce spikes again in the retinal ganglion cells. So we did similar first in mice, and then we moved to the primates. So you see here, for instance, we have injected the AAV vector in the eye of living primate, wait several months, and you see the expression here of catch, not channel rhodopsin, but catch, modified channel rhodopsin in the macula, because it seems that the expression using gene therapy is much easier or facilitated in the macular area. So what we did then was to take the retina of this primate, put it on a multi-electrode array, stimulate it with light, and you see that we have a high increase in spiking frequency in the ganglion cell at the macular area. You see this spiking frequency, and you see that it's really related to the position here of the expression of catch. So we do have the ability to drive the ganglion cells. So in this case, we were using catch, which has a peak of sensitivity around 450 nanometer. But this is quite toxic. It's blue light, which can be quite toxic to neurons. So we moved to another cells. So you see here, I just did some measurement of light toxicity, and this was on photoreceptors, but you see that it's blue light is highly toxic to photoreceptors, and it's why we moved to a red-shifted opsin. So this red-shifted opsin is Chrimson, and so we tested whether because at first we used the Crimson TD tomato. So we first tested whether Crimson alone or together with TD tomato was better. And we also tested whether AV2 or AV2-7M8 was better. And as you see, we selected this construct with AV2-7M8 and Crimson TD tomato at the end of the experiment. So we did again all the experiments in primate, and you see here the response of the retina, the macular area, to a sliding bar, and you see that it's highly related to the moving bar. And in this case, the movie is slowed down 4 times so that you could see the activity. And what is also interesting is that we tested different duration of stimulation, and you can see that the peak of the response is around 20 to 30 milliseconds, which is quite nice because it means that we can have video rate stimulation for this type of optogenetic therapy. So we wondered whether patient could be seeing symbols. So it's why we tested here different symbols. And we could show that it's possible to define symbols and that in fact the visual acuity that we might get with this optogenetic therapy is 20/249. And in this case, in fact, this visual acuity might be even decreased by the spacing of the electrodes. So then we wanted to define whether some signal was arriving in the visual cortex. So this is the response in a rat to stimulation, and this is the visual evoked potential. And you can see here the response and the long delay for the response. And when we did optogenetic stimulation of the ganglion cell in the eye, you see that the response in the visual cortex is much faster, simply because we remove all the phototransduction cascade and all synaptic transmission in the retina, so that in fact the response is much faster, not only as a VEP, but also when we record the spikes in the visual cortex, as compared here to the natural response. So we did also this in primates. So what we did was to record the natural VEP, so in black here, in an eye with Crimson TD tomato. And when we block synaptic response, you see that we have a very fast response, a small response, but very fast, exactly as in the right. But if we do the same test on the other eye, which was not injected with the AAV, you see that we do not see the same very fast response. And you see that this fast response is highly dependent on the light intensity used to do the stimulation. So we do have a signal that is arriving in the visual cortex when we stimulate with optogenetic therapy. So then the idea was that the intensity that we need is very high. So this means that we need to project in the range of sensitivity around 600 nanometer an image on the retina of patient. So it's why we generated some goggles. And you see these goggles with a camera here. And here there is a projector that will project on the retina the image in at 600 nanometer. So this is fairly similar to what was used for the prosthesis at the beginning. And but in this case, in these goggles, we use the camera that is not making images at defined time points, but a specific sensor that in fact record change in light intensity. And you see each time it crosses a threshold, there's a kind of event or spikes that is generated at that pixel. So this means that the pixels are only active when they are detecting change. And this is the kind of information that you're getting from these sensors. So only the frame of the object. And you can also generate a black and white image, because as soon as you have an event, you can also measure the light intensity to actualize this image. So with this type of camera, what we did was to show that we can model the activity of all the retinal ganglion cells at the millisecond precision, because these cameras are very precise. They respond at the microsecond. So it's possible to use them and in fact calculate the videos that are sent to the brain from the retina. So using these goggles. We have moved to clinical trials, and in fact, few weeks ago, there was a paper in Nature Medicine with the first patient seeing with optogenetic therapy. Let's see here some results of this clinical trial. I apologize, it's a So these are real, very recent data. So there's really a learning phase for this patient. This patient was blind for more than 10 years. And so there was real you see even a very light object can be seen. And so just finally to objectivate that there was really some performance with the seeing the object, EEGs were also recorded to really define that there was activity in the visual cortex when seen. So we showed that it's possible to recover vision in blind patients having lost photoreceptors. What we would like to do is not to move to patients losing their retinal ganglion cells. So this is the case for patients with glaucoma or diabetic retinopathy. So if they lose retinal ganglion cell, they lose the complete connection from the retina to the brain. So the idea is then for this patient to stimulate the visual cortex. So there are already some cortical prosthesis, and but we thought that it might be interesting to investigate other approaches. And so first, because we want to measure the activity in the visual cortex, and especially soon in primates, we have also implemented in the Vision Institute a new technology, which is called fast ultrasound functional imaging. So, and we tested whether we could see the cortical columns, the ocular dominance columns that are seen in the visual cortex. So these are the studies that were done in the 80s using deoxyglucose to show this ocular dominance column. And you see that they can be seen in the superficial layer, but not here. And so we did similarly using this ultrasound imaging. And you see that we need to generate a craniotomy to have the probe. But what is quite interesting is that we see also this ocular dominance column here in the visual cortex, as we could see here in histology, but not in this layer, of course, as shown here. And what we could show also is that we were able to generate a retinopathy using this technology. So we moved to apply optogenetic therapy in the visual cortex. So as you see, we have started with rats first, and we can express Crimson TD tomato again in the visual cortex. We have some neurons that are nicely labeled. And then we did some recordings, and we can see that we have some neurons that are sensitive to a visual stimulation, but also to the optogenetic stimulation. Again, what you see is the latency of the response is much shorter with optogenetic therapy. We can see that it's possible to activate neurons even very deep in the visual cortex using penetrating electrodes. And this is also the case when we do ultra-fast ultrasound imaging. We can see that we activate the visual cortex very deep using this strategy. And you see here the response with this technique. And we see that we can also increase the activity with increasing light intensity, just as we can see with the electrode recording. I think I'm done. So the conclusion I would like you to remind is that so it's possible to restore vision in blind patients with either retinitis pigmentosa or age-related macular degeneration, that the best visual acuity is currently generated with the primate retinal implant, and it's close to threshold of legal blindness. We have an alternative solution with optogenetic therapy. In theory, we might get a better visual acuity. We got the first response with optogenetic therapy in a patient having retinitis pigmentosa. And so now our future project now is to develop visual restoration at the cortical level. So all this was generated with all the people at the Vision Institute. In my team, especially Grégory Gauvin for optogenetic therapy, Paul-Henri Prévost for the retinal prosthesis, and Kevin Blaise for ultra-fast ultrasound imaging. This was a collaboration with Denis Dalcara for all the AAV technology, Jens Dudel for some patch clamp recording, Riyad Benosma for the recording and stimulation, and of course José Sahel, who was the head of the Vision Institute, and the company GeneSight and Pixum Vision. So GeneSight for optogenetic therapy and Pixum Vision for the retinal implant. And other colleagues. Thanks a lot.
Okay, thank you very much. So we can all thank our speaker. Thank you very much. And we have some time for questions. Are there any questions? Okay, maybe Serge, I have a few questions. First, again, thank you very much for sharing with us the optogenetics results, both in the primates and in humans. And you said that there is some theoretical limits to the visual acuity of what you show in the in vitro studies. And in the human, it is fascinating, but still the results are not that great. And what do you think are the limitation or the challenges with this approach? This is one question. And the other question is, what are the advantages of using the event-based goggles over other projection system like Pixum is not using the event-related or event-based goggles? So it's two different questions.
So first, for the question about visual acuity and in patient with optogenetic therapy, it's clear that we are with a patient who was injected with the middle dose. Patients are now injected with higher dose. Unfortunately, due to COVID, these patients were postponed, and so it's too bad. But we hope that with higher dose of vectors, maybe the result will be better. Then this patient was blind for more than 10 years. So maybe also the recovery is highly relying on the years of blindness. So maybe the performance would be better with a patient with a more recent blindness. And then, in fact, when we measured the visual acuity in ex vivo, I mean, it's always difficult because we extrapolate this from the recording of all the retinal ganglion cells, so which is a mix of ganglion cells. So it might not be as good, and also we might be lowering the measures because of the spacing of the electrodes. So we don't exactly know. We did this with two different approaches, either with the symbols, but also with some kind of flicker stimulation. So, I mean, we still need to have the response from patients because they will really provide the information. Then concerning the camera that is used in the goggles, the advantage of the event camera is that it provides immediately the contour of the objects. So there's no need for calculation to get this. And in fact, it's quite interesting to decrease the level of light that is shining on the retina because it's quite intense light. And also, we don't know, I mean, with time, what will be the effect of the stimulation on the opsin. So it's better to reduce this light intensity as much as possible. So this is one thing. The other advantage, the great advantage of these camera is that they have no, they cannot saturate. So even if you watch the sun, they do not saturate. So the patient is not knowing what he's watching, if he's watching a light source or the sun or anything like this. So in any condition, this camera will work. This is an advantage as compared to classic video cameras that will not be able to work in all conditions. So this is also a great advantage of this camera. At the beginning, their resolution was not as good as video cameras, but now you have a VGA quality, and also the quality will further improve.
Serge, I also want to ask a question if possible. So first I want to say thank you for an almost a science fiction talk from my point of view. Very fascinating. I want to ask, so I understand that the temporal resolution, regardless of the spatial resolution, is quite good. Even if the implant doesn't have a great resolution or the camera. So I wanted to ask, what do people that experience this report, if when they actually move around, not when they sit and they have to see what object is in front of them or to read something, but rather when they walk around in the environment or move around, even in a room that is safe. What do they report? Do they say that the motion, that the movement provides them with more information? Do they feel that this is sufficient for them to move around and act, or were they asked about these?
I'm not following the clinical trials, so I would not be too easy for me to answer. But I think some patients said that they can easily recognize, for instance, the patient with optogenetic therapy, he said that he was able to watch the crossing for the streets he was able to distinguish them and to easily find them out. So then what is quite interesting for the patient with the age-related macular degeneration, and this is what Yossi had shown with the animal, but what is very interesting is that they can fuse the artificial vision with the natural vision. And this is very important because they can establish a continuity between the two images. And they fuse the information so that it might be very interesting because they might get the color from the natural vision, the shape from the artificial vision. And it's quite interesting. I think all the studies that will come out now will be very interesting to see how, in fact, they can really use this type of artificial vision. But I think it's really a start, and we will really learn from the patient. So.
Okay, thank you.
I have a question
one
too.
question.
Oh,
Can I go? Okay. So, Serge, a few questions. So first, you said that you block the light responses from the photoreceptors to isolate the responses activated by the Chrimson. How do you block photoreceptors inputs?
Yeah, yeah. Sorry, I'm sorry, I did not mention that. But of course, when we did also the studies ex vivo, we were blocking the natural response of photoreceptors by adding synaptic blockers in the baths.
So this
so
was
that
done
we would
on
be sure that what we were recording was simply the optogenetic response and not some kind of natural vision. And this was much more complex in vivo, because in vivo, I mean, it was very difficult to be sure that we were removing all the natural response. And it's why we injected in the eye the blockers, the synaptic blockers, so that we could remove all the natural activity. But this was, I mean, it's always very difficult to be sure that you have completely removed all the natural responses.
And do you know how the virus affects the different neurons, both in the retina and in the visual cortex? Meaning, for example, we see that natural stimulus with photoreceptors will activate both on and off responses. And here, obviously, you only have on cells and off cells that are activating in response to the light onset. Do you think that in the future you will be able to direct to specific cell types?
Yeah, this is clearly the objective, is to try to have more specific stimuli. And this could be done using optogenetic therapy, because when we will get some specific promoters, we will have the possibility to target one cell type, and then it will be much more specific. Because in this case, currently, we express Chrimson in all types of ganglia cells. So it's clear that both on and off are all targeted. So, I mean, it's very, it could generate some strange activity in the brain when both on and off cells are activated at the same time with the same stimulus. I mean, it's clear that this could also deteriorate the performance of the patient. It's clear. But we have no specific promoters at the moment. So.
Thank you.
Peter, did you have a question?
yeah, but it was the same as the, I was going to ask about the specificity of the ganglion cell type stimulation, whether, you know, what would be the technical difficulties in achieving this. And so Serge has already replied. Yeah.
Well, it's like the implants, in fact. When you're using implants, we don't know which cells you will be activating. So I think it's fairly similar. And what was very impressive was that, in fact, the patient can perform even with this type of confusing information.
Yeah, no, I agree. It's impressive that they can decode such a I mean, it's incredible that you can do this at all. Let me just say this, but given that, it's also surprising that with such coarse cell type stimulation, you can achieve some vision. So, yeah. Thank
Can I
you.
just ask one more question then, Serge? What's the situation with people that are blind from birth? Is there any chance of activating their visual responses by any of these approaches?
Well, this is kind of the dream, but we are not yet there. I mean, first, it will be easier with patients who have already seen, because in fact, their visual cortex was kind of developed for seeing. I mean, we all know the work from Wiesel saying that it's not possible to see if you have not seen during your infancy. But recently there were some studies showing that if you are operated from cataract, when you have congenital cataract at a very young age, when you at birth, you don't see and you never learn to see. But these patients who were operated even after being adults, they kind of recover some vision and they learn to see at the adult age. So if they can learn to see, it opens a new window of possibilities for the patient with blindness at first. But this is again something, we should first, it will be easier with patients who have already seen, but then we should be open-minded for all the patients.
Thank you.
I have a question. First, thank you for a fascinating talk, Serge. And I have actually two questions. The first one is, do you see any effects of eye movements that the patients or animals are making on the evoked responses? Either, most probably, the best location probably to look at is in the cortex. This is one question. And the second question is about plasticity. So these implants are planned to work for many weeks, months, and probably years. And there were several studies showing that long-term stimulation causes changes in circuitry, in synaptic strength. And I was wondering what kind of effects do you think this will have on the future development in this field?
So maybe, well, it's quite difficult to know, but I think for the moment, the implants are quite stable, and also, for the retinal implant, because these implants are not penetrating and they are not moving with respect to the retinal tissue, they might not affect the performance, because they might be really long term. If they are not degraded by the tissue, they should perform quite well. And especially in the case of the primate implant, it's really a small chip of 2 by 2 millimeter, so it's very light, and so it should not move relative to the retina. So this high stability should somehow preserve the implant. The fact that it's outside of the retina, there might not be too many cells to kind of digest this implant. So I would think that this should be quite stable. And up to now, we have not seen any change. We also used it in primate for very long, and it was still stable and working. So I think I mean, I'm not seeing too much problem. So we're thinking now to check also the activity in the visual cortex when we activate this implant, and to see how we, even in primates, what kind of information processing or activity we can induce with this implant. And we're thinking also, I should have mentioned that all the optogenetic therapy was highly developed with Bottom Rosca. I mean, we highly contributed, and he was there recently. So yeah. I guess it's our thoughts kind of overlap.
Okay, so I think that in terms of plasticity, what I meant is that when you do microstimulation and you activate multiple neurons that previously were not supposed to work together, and this has to do with the fact that the stimulation is pretty coarse relative to the physiological activation, then actually you generate activation within a circuitry that is different than the, if you wish, the normal one or the one that usually the animal is using, and this may cause changes in weights of synapse, which may lead to additional changes. And I was wondering about this on the long term. And also I wanted to know if you see any effects that are related to eye movement, because we know that there are effects of eye movement like saccadic eye movements and microsaccade in the visual cortex on neural activity. So I was wondering whether you see any reports of the patients regarding the phosphene or the object when they're making eye movements, and whether the patient are at all changing their sort of like eye movements in the blind when you start the stimulation.
So I mean, for plasticity, I think it's what we see is that there's really a long learning phase. So this might be relevant to this plasticity because I mean, what has been also described with August was that in fact the patient, before the implantation, if you just flash light, they have almost no response in the visual system. But after using the implant for 1 year, if you shut down the implant and you do the same flash of light, there's a higher activity in the visual system. Meaning that in fact, there was some plasticity to kind of use as much as possible all visual information that are arriving to the visual system, as if the system, the brain has understood that visual inputs are containing against some information, and they have to be processed. I mean, but I think it's very interesting what is there. And so what we would like to do is really to follow patients during the course of this learning to see, in fact, if the same image has not different type of activity in the brain at the beginning of the recovery and later following the recovery. So this is quite important. For the eye movement, I would not know too much, but what you have to realize is that in both cases, retinal implants and optogenetic therapy, the active cells are just at the macula, and also you have a video projector projecting an image. So this means that if you want to see with this type of artificial vision, you need to watch straight in the goggle because you need to have your fovea on the axis of the video projector. So it's clear that I mean, the patient has to learn to keep its eye straight in the video projector. So, I mean, then is he doing some kind of eye movement? I don't know. And but it's clear that he has to kind of keep his eye as much as he can straight in front of him. I'm not sure I answered your question.
Yeah, I think what I meant mainly is that we know there are extraretinal effects in the visual cortex that originated from eye movements by themselves. And usually normal humans and primates, there are about 2 to 3 microsaccades or saccades that are done per second. And these influences are integrated with the visual inputs and that are arriving from the retina. I was wondering whether you see some effects of this eye movement in relation to the phosphines that are generated by this artificial stimulation.
Yeah, I don't think we went so far into the understanding of what's going on.
Okay, thank you.
I can add that there was a paper by, I think it was Renner, the German group that was doing retinal prosthesis, and they've shown that there is a decrease in visual eye movement when the chip is on. It means that it is helping to fixate the eye on the picture. As however, with what you are asking, Hamutal, so people do think that when you are moving the eyes, then you have more visual input into the retina, and this might also happen in prosthetic vision. It's difficult to show it, but it can also contribute if it is connected with the optics of the projection system. For example, with second side, where there is an external camera, then moving the eye will not help. Then they have to move the entire head. And I think that in the video of the optogenetic guy that was transplanted, he was moving the entire head. He was doing like this.
Yeah, it was just like trying to scan, I think, the visual scene or something like that. Yeah, that would not be a typical eye movement. Yeah.
But it's a very important question.
Especially given the recent thoughts in the past decades suggesting that vision is actually generated also in an active manner, which involves the eye movement. So I was wondering about this. Thank you.
But it's clear that if the projector is just in the axis of the eye, they will rather move the head than the eyes.
Okay, maybe a last question, anyone? Okay, so we can thank again our speaker for this exciting talk. Thank you very much, sir, and we are looking forward to see more results next year, maybe. So thank you all, and see you in our next
seminar.
Thank you. Thank you very much for the invitation.
Thank you very much.
Bye-bye.